Assalamu alaikum, dear students. Today we are going to discuss a brief structure of the HIV virus, and we will also be discussing the life cycle of the HIV virus. I'm going to upload short lectures so that one concept at a time should be shared with you people. So here, what do we have? Just before starting the life cycle, a general structure of the life cycle of the structure of an HIV virus. Human immunodeficiency virus HIV. You can see that it is made up of different parts. You might already have studied that generally a virus, uh, it consists of an inner core which is going to cause the infection and an outer protein part which is known as the capsid. Among the most important characteristics of a virus is the nature of the proteins. ठीक है? It is the nature of the proteins that make up the capsid. And it is these proteins that determine the infective properties of most viruses. However, we have some viruses in which there is an other covering except the capsid. I have just told you that the capsid is made up of proteins which determine the infective value of the viruses. Now, in some viruses, especially in human viruses, we have an outer envelope. This envelope is made up of special chemicals, of special molecules, biomolecules, which are known as glycoproteins. These glycoproteins, they make up the envelope and they make up the spike. The envelope of the HIV virus is mostly acquired from the host cell membrane. It has a very similar structure to the host cell membrane, and that is why it kind of cheats the human immune system. When an HIV virus infects humans, it goes into the bloodstream, and most of the time, it is always sexually. The contamination is transferred either sexually or most of the time through a hypodermic needle. हाइपोडर्मिक नीडल का मतलब ये होता है जो स्किन के नीचे जो इंजेक्शन लगता है. The virus particle, after it enters the body, it circulates throughout the body, but it does not infect most of the cells that it encounters. Why? It does not infect most of the cells that it encounters because it needs a cell where there are specific receptors, and it is those receptors where it is going to be compatible and it is going to enter that cell and then it is going to infect that cell. For the HIV virus, the cells that are carrying the receptors which will accept the virus and which will give the virus a pathway to enter are actually the white blood cells, and specifically the CD4 lymphocytes. In the CD4 lymphocytes, this virus attaches itself by these glycoproteins. These are two glycoproteins. One is this one, and one is this one. Glycoprotein 120, glycoprotein 41. They have different functions. This glycoprotein, the function is attachment, and this glycoprotein, the function is fusion with the cell. This is the envelope of the HIV virus, which is acquired from the host cell membrane. Inside the HIV virus, when we go inside, we see this protein core, which is made up of various protein blocks, which are called the capsomes. And inside the core, we have the RNA molecules. These are the RNA strands. We have two RNAs, two copies of RNA present, and we have three enzymes present inside. These enzymes are not active inside the viral body. However, when the virus attacks the human body, these enzymes, they become active because they start to share the biochemical machinery of the human body. ठीक है, तो अब इसमें हमारे पास कौन कौन से एंजाइम्स हैं? रिवर्स ट्रांसक्रिप्टेज, इंटीग्रेज, protease. These enzymes, they have specific functions. रिवर्स ट्रांसक्रिप्टेज का फंक्शन क्या है? रिवर्स ट्रांसक्रिप्टेज का फंक्शन है कि वो वायरस को जो है, इट कन्वर्ट्स दिस single stranded RNA into a complementary DNA, which is then going to attach with the DNA of the human cell. Integrase. Integrase helps to integrate the complementary DNA that has been produced with the human DNA. And then protease. Protease helps to break down the larger protein particles. Okay. Now, when we study the life cycle, we will go through the detail of the life cycle. This was generally the structure of the virus. Now, let me share another image with you people so that you can see the structure of the virus very clearly. Okay. 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 First of all, now, after going over the structure, you have to learn the parts of all the structures, and then you have to go over the structures, because in when we are studying the life cycle, we have to be very clear about the different parts of the viral particle. Okay. And when we see the structure of the viral particle, I will be showing you this life cycle. This life cycle is very good so that you can understand it, the life cycle, and you can draw it in your paper. Now, this is the first part, the fusion and entry of the HIV virus. It comes inside the HIV RNA. This is just a brief summary. Then, secondly, this is the action of the reverse transcriptase, which is converting the HIV RNA into the HIV DNA. Now, this HIV DNA, this is the second enzyme. I'm just showing you this slide, so you should be very clear about the function of the enzymes. This viral integrase is going to integrate the viral DNA into the human DNA here. And then after that, this viral DNA, it is going to be copied in the form, like we say, the transcription takes place and messenger RNA is produced. The viral RNA is going to be produced. This viral RNA is then released from the nucleus, and the viral proteins are going to be produced and they are going to be organized. And then after they're going to be organized, that phase is known as the maturation phase. So we have the fusion and entry phase, we have the reverse transcription phase, we have the integration phase, we have the construction phase, then we have the maturation phase, and we have the budding off phase. Okay. In the next lecture, now I'm going to upload the second lecture. We will be studying the life cycle of the viral particle inside the human body and how it affects the human body. Okay. See you people in the next lecture. Love is over here now. I'm going to